Hello, everyone. My name is Danielle Roberts, and I'm a biology teacher here at Beijing Huijia Private School. When I introduce myself to people and tell them what I do for a living, I often get some variation of, "Oh my God, I loved biology in high school. Learning genetics was so cool." They might even bring up the old Punnett squares that we use in our biology classes still today. Or they might tell me how much they love learning about the immune system and white blood cells that are capable of gobbling up pathogens for breakfast and keeping us safe from disease. Or they might even mention ecology and how much they love learning about ecosystems and the interconnectedness of all the organisms within it. That can get them excited about talking about other aspects of their science classes that they remember, talking about chemical reactions in the chemistry lab, and talking about learning forces in physics by pushing things off of high places or riding roller coasters in a theme park. This discussion creates a nostalgia in them as they remember the curiosity and discovery that they felt in their science classrooms. And as a science teacher, I love hearing about this. It's what inspires me in my own job, that I can help students discover their love for my subject and the world around them. But then, I actually start to consider what they remember about science. And it becomes clear to me what makes these experiences memorable to them. These experiences brought about a shift in their thinking. At one point, the evidence from their senses told them to understand something in a particular way. But upon gathering more knowledge and more evidence, they shifted their understanding. Take the example from physics of a feather and a bowling ball dropping in a vacuum. Our own experiences tell us that the feather should fall much slower than the bowling ball. However, our physics teachers insist that if we put these two things into a vacuum, they'll fall at the same rate. And then our physics teachers give us the evidence. They show us the experiment, and the bowling ball and the feather miraculously fall at the same rate. How can that be? We now have new evidence, and we must rearrange our prior ideas to fit with this new information. Each of us has undergone our own small paradigm shift. In case this phrase doesn't sound familiar, let me elaborate. A paradigm shift is an idea developed by the philosopher and scientist Thomas Kuhn in the mid-20th century. He used it to refer to a fundamental change in the accepted theory of an academic discipline. A good example of this is the development of germ theory in medicine. For most of history, we didn't and couldn't know what caused diseases. We had little ideas about microbes and pathogens, so we looked for evidence elsewhere and made the correlation that a bad environment must be the cause of diseases. The recommendation of leaving the city air and getting out to the seaside or the countryside seemed to help sick people, so that idea seemed reasonable. Then, the father of microscopy, Leeuwenhoek, started to put anything and everything he could find under a microscope. And we began to understand the living world that existed below our own eyes' resolution. Eventually, biologist Robert Koch took these ideas and suggested that there was a cause and effect relationship between the microbes and the diseases that caused them. Thus, germ theory began to gain acceptance. This is an example of a paradigm shift. And much like education forces us to do, when we get new evidence, scientific disciplines have to go through these earth-changing shifts. That's what pushes science forward. That's what is essential for the growth of the discipline. But in school, we don't often learn about that bumpy road that it takes to get to a new understanding. A lot of paradigm shifts are taught in science classes, and they follow the same pattern: a sudden and seismic shift from one way of thinking to another. Think about the story of Isaac Newton. sitting under his apple tree, an apple falls on his head, and bam, he understands gravity. Or Archimedes trying to figure out how to determine the amount of gold in an object while taking a bath, when suddenly, eureka, he has the idea of using the displacement of liquid to identify the metal inside the object. These ideas make great stories, but the problem is they're just stories. They don't include the hundreds of scientists that have come before, or the hours of testing, hypothesizing, revising that went into the development of these paradigm shifts. In reality, it's much less similar to Newton and the apple, and much more similar to what Newton himself commented about his own progress. If I have seen further than others, it is because I stood on the shoulders of giants. Paradigm shifts are slow, winding processes that are built on previous scientists' work. I would like to show this today with my personal favorite paradigm example, yes, I have one, of DNA as the genetic material. The road to the acceptance of DNA as the material that made up our genes and passed genetic information was not smooth. I remember in high school learning that Dr. Watson and Dr. Crick were the first people to discover DNA. That they told the world, this is DNA and DNA is the genetic material. The days before, scientists were still arguing, what is the genetic material? Is it even DNA? And then suddenly, textbooks were proclaiming it as settled knowledge. And teaching it in this way leaves out so much context that is important for accepting science in today's world. At the beginning of this story, everyone understood the general phenomenon of heredity fairly well. We had seen that children look like their parents. Farmers had managed to breed livestock and crop plants that had certain traits. And we had also managed to make a variety of dog breeds that had vastly different characteristics. So we understood that there must be something passed from parent to child. Most scientists thought the answer lay somewhere in a molecule found in our cells. As a biology teacher, let's go back to biology class for just a minute. As we all might remember from our time there, our cells are made up of many different biological molecules. Of particular interest to us are proteins and DNA. Proteins, like those behind me, are made up of small pieces called amino acids. These 20 amino acids can combine in any sequence to create practically infinite number of proteins that do the cellular work of life. These amino acids and proteins, they were first discovered and investigated in the late 19th and early 20th centuries. Because they can be such complex molecules, scientists felt pretty confident that they were at least related to the ideas of heredity. Perhaps it was proteins that were passed from parent to child. But before we could say for sure, we needed more investigation. Now, DNA. DNA is a combination of just 4 nucleotides. The scientists who discovered DNA even noted that it seemed like a pretty simple molecule with just these 4 building blocks. Biochemist Phoebus Levine even put forth something we call the tetranucleotide hypothesis. This is the idea that DNA was made up of mostly of these 4 repeating bases. And each unit of DNA repeated itself, meaning that the bases had to be present in roughly equal amounts. This hypothesis was widely accepted by scientists and led scientists towards the belief that proteins must be the unit of heredity. DNA is just too simple of a molecule to make organisms. Now, you might not know this, but I'm going to let you in on a secret. Scientists love proving something wrong. I always find it funny how much people think that we want to support a hypothesis. It's far more interesting to us that we can be contrarian and reject a hypothesis. Oswald Avery was just this kind of scientist. He was working away in his own lab trying to determine the unit of heredity. He, like other scientists, had narrowed it down to either proteins or DNA, but he just wasn't convinced that it was proteins. He used different strains of bacteria to prove his point. He grew these strains together, removed essential elements from them. either the DNA or the protein. If one could morph into the other, that meant that that missing piece must be the unit of heredity. Removing proteins from one strain and mixing the two strains together did not cause them to become each other. But removing DNA did. Strain A morphed into strain B. Because strain A lacked that essential DNA to recreate itself. The search for heredity had begun to unravel. Then, along came my personal favorite scientist, Dr. Edwin Chargaff. Chargaff was a biochemist, and he had a special interest in the nucleotides that made up DNA. Avery's conclusion that DNA could be the unit of heredity didn't fit well with the tetranucleotides hypothesis and the claims that DNA was just repeating units of the same thing. And this contradiction intrigued Chargaff. See, I told you we're contrarian. He began using chromatographic techniques to study the amounts of nucleotides in the DNA. And he discovered something amazing. The 4 nucleotides were rarely, if ever, present in the exact 1-to-1-to-1-to-1 ratio that would be required for the tetranucleotide hypothesis. Instead, he showed that in almost all cases, the nucleotides A and T were found in about equal amounts, and the nucleotides C and G were found in about equal amounts. This suggested some relationship between the two, but Chargaff stopped short of getting to that conclusion. Nevertheless, his findings pushed even more scientists to reconsider what was the unit of heredity, and he was a major stepping stone for Watson and Crick. Finally, we come to the ideas of Hershey and Chase. These two are contemporaries of Watson and Crick, the ones credited with the discovery of DNA. And Hershey and Chase used viruses instead of bacteria. Viruses are composed simply of proteins and DNA or RNA. Viruses inject their own genetic material into cells and use the cells to make their molecules for them. So, if viruses are injecting proteins, then proteins must be the unit of heredity. But if viruses are injecting DNA, then that must be the unit of heredity instead. But how to prove it? Well, Hershey and Chase decided to use radioactivity. They tagged proteins with radioactive elements in one set of the virus, and they tagged DNA with radioactive elements in the other set of the virus. If they found radioactivity inside cells infected with the radioactive protein virus, it can be concluded that the proteins made it inside, and were thus the unit of heredity. And similarly, if the radioactivity was found in cells infected with the radioactive DNA virus, then DNA made it inside, and it must be the unit of heredity. When they analyzed the results, they discovered, just like Avery, that DNA was being passed down from virus to cell to the next generation of viruses made in that cell. Proteins were necessary for cell function, but not what moved between the generations. All of these scientists and many others were all aiding Watson and Crick. The ideas of Watson and Crick were shaped and molded by this gentle shift in their field. They used the ideas of others, just as every scientist has done since the first person systematically tested a hypothesis. Watson and Crick used these published papers in their race, but I also want to mention another scientist, Dr. Rosalind Franklin. Rosalind Franklin was the scientist behind the X-ray crystallography image that we see in photograph 51. Okay. See, Franklin was another scientist working at a rival lab that was racing to find out the answer of what DNA was. She was a master of X-ray crystallography and was maybe the first person to recognize the helical structure of DNA. This photograph that she took was obtained by Watson and Crick without Franklin's knowledge or proper attribution of her work. It was one of the final pieces of the puzzle that helped Watson and Crick to crack the code, so to speak, on the structure of DNA. Watson and Crick made headlines around the world, and their names became synonymous with DNA discovery, while many of these other scientists remain in the history books. And now we're all familiar with Watson and Crick's amazing discoveries. It's something most high school students don't have to think twice about when asked. But when we learn about it, we can easily categorize it into a eureka moment, instead of recognizing the long slog full of stops and starts that got us this discovery. So, why journey here in this discussion? I really, really like talking about DNA, but that's not why I talked about this one. I could have talked about so many other discoveries in biology or any science that are framed in this way. A scientist worked really hard, had a moment of inspiration, lightning strike moment, and then a new discovery was made and accepted. But that's not how it works. Science is collaborative and it makes mistakes. Before, these mistakes could be kind of hidden from the public because it was impossible to publish every small detail of every science in every newspaper around the world. You could only get this knowledge if you sought it out, and to seek it out, you were generally very enthusiastic. The general public, on the other hand, didn't get this knowledge until it was considered acceptable. If we look at the discoveries in science happening in the modern day, it's different. We have all of this information available at our fingertips. If we want to read an article, we can go online and find it. If we can't, our wonderful librarians can help us to get that article. Scientific communicators help us interpret these online. The polished veneer of science in the past is no longer there, and we can see the rougher, uglier side of science. Being so close to those rough edges all the time has made some people suspicious of science in general. I remember when I was younger, my father was frustrated with nutrition science he would read in the news. He said one week the news was telling him that coffee would kill him, and the next week it was telling him he needed to drink 4 cups of coffee to stay healthy. The rapid pace and change and conflicting ideas can make science seem less reliable to some people. But we need to recognize science for what it is, an imperfect way to describe the world. Science is tested, verified or rejected, and the process starts again. This is a feature of the science and not a bug. And I hope that an emphasis on the kind of science education that shows the dynamic and turbulent nature of discovery can help us combat the distrust in science as we move toward a future even more dominated by science on the edge of knowledge. Thank you, everyone.